we're so big, it's difficult to know what other people are up to. And we have this tendency to, live in these silos. We got to figure out a way to bust through those. And so what we're doing here is, in breaks in the visitor colloquium schedule, what we're going to try and do is fill them in with, individuals from inside U of T astronomy to give them an opportunity to tell you guys what what exciting stuff is going on. Now, I think this is a stopgap, okay? Because we used to have this G2000 thing that where it was give us kind of a flexible opportunity to do this kind of thing, and for some reason I haven't quite figured out that's gone. But we need to capture some of that back, and so this is an early attempt to do that. So what better way to do that than with two superstars from the department to give you a talk? Okay, so our two superstars are Renée and Ting, and really, I guess I'm not gonna embarrass either of them with a huge introduction, other than to say, well, I guess Renée's been around for a short period of time, and Ting has been around for an even shorter period of time. So if you don't know either of them, then you are missing out. So you should figure out a way to introduce yourselves to them and ask them about what they're up to, and particularly for the students. It's a great opportunity for you to talk to people who are doing really exciting stuff, and you should avail yourself of the many opportunities to talk with great people at U of T. Anyway, okay, I will stop there and just pass it over to Renée, who's going to talk about, wow, this title just blows me away. Can astronomical methods help detect signs of neurodegeneracy? So Renée, take it away.
Cool, thanks so much. I should say, so this is in collaboration with colleagues of mine at University of Laval in Quebec. And the unsung star of the show actually is Bolin Van, who did a 1500 project with me over the summer. And so this is a lot of our collaboration, but a lot of his hard work and sweat and tears. So make sure to keep talking to him about this as well. But it really started off as I went to a meeting in July of 2020, and it was funded by CFAR. And the idea was, can we get biologists and astronomers to just talk about what they do? And so I gave an overview talk about some of the cool machine learning and astronomy stuff that's done by, some members of our department, even people like Kristen and others, and the kinds of things that we care about in astronomy. And there was a woman in the audience who gave a sort of similar talk about the work that she does in partnering with machine learning experts. And at the end, she came up to me and she said, your data looks like my data. Why are we not working together? And so we decided, initially we wrote a small little grant to just fund the initial thinking about this. There are actually 3 or 4 different wings to our project. And then based on that, we decided to get funded from a larger initiative. And so this is now funded by both CFAR and the New Frontiers in Research Fund. And it just came about because I gave a talk and we looked at each other's data. So don't only go to talks in your subject area. Okay, so what's the backstory? So I am not a biologist. In fact, I did not do a lot of biology, even in high school, but I'm interested in how astronomy methods can transfer. And so the crux of the matter is you have axons and in your neurons, and signals are being transferred between the terminals or the ends of your neurons. And those signals are responsive, those neurotransmitted signals, or the synapses are telling your brain to do different things, and they're firing in different ways depending on what activities you're doing. The colleagues of mine that work in biomedicine are particularly interested in how those firings change in people that have various neurodegenerative diseases. So specifically, they're interested in ALS and Parkinson's. And the way those transmissions happen changes if you have those diseases. So how do we actually measure how those things happen? Well, there are 2 different things that you need to pay attention to. The first is you can talk about the strength of the synaptic interaction, how energetic it is, or how bright it is in this case. And you can also look at the distribution of proteins, the synaptic proteins that are responsible for mediating those synaptic neurotransmissions. You can actually look spatially for how they're distributed along the ends of this axon terminal, between off the basic structure of the neuron having these synaptic transmissions. Our work is actually going to look at both of these, but for this talk, I'm just going to concentrate on the sort of brightness of these interactions. Can we detect sparks of neurotransmission in an image? So it's actually a nice, simple project. And you might say, well, how do we even see those neurotransmission events anyway? So the data that we work with is these are cells taken from the hippocampus of mice. And if you want to see these synaptic responses, what you do is you actually realize, if you're a biologist, that one of the chemicals responsible for mediating the reaction is calcium. And so what they do is they add to these hippocampal cells, they add a dye that is sensitive to calcium. And so the dye is telling you when calcium events are happening, and you're using that as a proxy for the events themselves, right? Oops. So, my colleagues at U Laval, they work on high resolution microscopy. So they have sort of 250 nanometer resolution, spatial resolution, and 150 microsecond resolution. And so they're able to make essentially what look like movies in these cultivated neuron cells. They often call them models. So if you hear me say mouse model, it's basically the mouse cells. And they can look for these flashes. Now, in an example of one of the slides or one of these sort of images, you see her in the bottom of the screen. So this is a false color image in the same way that our work is false color images. And the brightness of the image is telling you the kind of concentration of calcium in that part of the image. And you can see this neuron structure. You've got these dendrites, these long filamentary arms, and then the synaptic activity is going to happen kind of off the dendrites, potentially kind of off and onto the dendrites. Okay. So why do we care about this? Well, you can take models of how the firing would be different in mice that are doing different things. So whether they are running or climbing or whatever, you would expect the patterns, the synaptic pattern response to be different. And other colleagues have shown that the different patterns look different if you have signs of neurodegeneration. The reason why this is critical is because the signs of neurodegeneration at the synaptic level are typically evident way sooner than the macroscopic signs of neurodegeneration. Once you exhibit signs of Alzheimer's or ALS or Parkinson's, it's much harder to treat that because the disease has progressed. But if you actually can detect it at the level of cells from your brain, the hope is that you can treat this much earlier. Now, of course, a lot of this is blue skies. The end stages of this is kind of blue skies research, but that's the goal, and that's where we come in. So what has already been done? So as I said, the talk that I gave my colleague Flavie Lavoie-Cardinal at U Laval, she's already working with machine learning experts. There are lots of AI chairs in the Brain Institute, the Servo Lab. And so what they do is they take these slices of images at different timestamps that have these filaments on them, and they apply a kind of thresholding analysis. Now, anyone who's an astronomer, already you can start to see that the light bulbs are going off. They then can feed these different thresholded analyses into a kind of machine learning approach and do better than just by eye, because the problem is if you're just using your eye, and if you're a graduate student counting all of these potential transients, that can be a very long time. So, in one neuron, you expect roughly 6,000 frames of these microscopic images, about 100 to 5,000 calcium transient events in a tiny little field of view. And so their estimates, and I should say that a lot of these slides are from my colleagues, their estimates are roughly 40 minutes of computational time per neuron, which if you really want to make statistical inferences is a lot. And so that is why they're working to develop these kinds of techniques to make sure that they do a better job of transient detection. So how can we help? I don't want us to be arrogant and think that they don't know what they're doing, but there are parts of the problem that assumptions are made in various fields that extend for long enough in the future that people just stop questioning them. And so we were interested in the tools and techniques that we use in astronomy to inform some of this work. So the first thing that we do is we do a lot of difference imaging, right? The top images are from the Deep Lens survey, and we typically take an image of a galaxy area that has been, monitored over a certain number of certain time to make a template. We then take an image the next night and we see there's a difference, and we use that to detect a transient, to detect like a supernova or something like that. And so the idea of difference image analysis is something we do a lot of. And similarly, some of my colleagues have spent a lot of work in looking for filamentary structures. So if you want to look for filaments of H1 in radio data, then you use something like the tools and techniques developed by Susan Clark and others to look for these thin filamentary structures and also to understand their position in kind of 3D space. And so I pitched this as a project to Bolin in the beginning of the summer, and I was really excited because he has a great combination of an interest in a lot of these methods, and also, the kind of excitement of trying new things that we haven't done before. And so we decided to look at a kind of pick and choose a list of things we could potentially do in astronomy. And the first thing we thought of doing is, how can we do better at modeling their background? Because they often have microscope configurations that change, so their data is not often very uniform. They can't always find the thresholding is kind of quite tweaked and not always optimized. And so we decided first to run the rolling Huff transform to do a good estimate of the background, and then we would sort of make a model of the transient-free background in any particular image, and then use difference imaging to find these flashes. And then we have to apply sort of kind of cleaning algorithms to detect all the different transients. So I'll talk through a couple of these different steps in our process. But the first thing to do is apply something to find the filaments, and before you can find the transients off the filaments. And again, it's tricky because the background noise of the microscope images are also changing with time. And so the rolling Huff transform is sort of like a way of finding sticks in an image. You sort of create a thresholded bitmap. So there is a little bit of thresholding that we haven't completely replaced the thresholding that's done by the biologists. But then you can look, sort of find the stick by rotating that length that you have, and finding not only the sticks, but the interfaces of the various sticks together. And so you can measure from one image, you can basically create a map of where you think the line-like features are in a particular image. And so this method worked pretty well. And so in this sort of 4-panel plot here that Bolin made, you can see both the original image at the top, and the sort of transient has been circled for your viewing pleasure. And then we have one model of the sticks themselves, which is in the bottom, and then one model of the dendrite-free image. So we wanted to map the dendrites or the sticks, but we also want to measure the background. So typically, if you just take the average of the whole image, you're going to get a bad estimate for the background. And if you just take tiny little patches, that's pretty computationally intensive, and you don't know where to draw the background, the boxes. So we wanted to use this to split up into dendrites and background. Once we could do that, we can make a pretty good estimate of the background. And you can see here that the transient that we're hoping to detect later in the difference image is gone when we measure this, the background. So the next thing to do is compute these difference images, and then find out, sort of cluster the individual pixels that we find as transients into groups and clusters. Because of course, if we just look at any pixel that is flaring, then you can imagine being sensitive to just a lot of noise in your image. And so a nice little thing about this is, as we were doing our research project, our research literature study, we actually found that it's already the case that astronomy methods have been used in biology. So in fact, the radio astronomy clean approach, where you basically take your point sources, you subtract the brightest one, then you sort of model the PSF, take off the brightest one, and then continue to do that in an iterative process. That we could adapt for the case of the Cancerian transients themselves. So this was a really fun part of that. And so what Bolin did is we asked our colleagues to image sort of like a beam inside their microscope to give us a sense of the point spread function, because in general they didn't necessarily think of doing that, but they measured a bead, a calcium sort of bead, and then that gave them a measure of the point spread function. And so what Bolin then did was use the clean algorithm that we have, which includes this point spread function, which is sort of related to like the diffusion of calcium as well as the image properties, to test how well we could remove simulated points that were close together. And this plot kind of shows that, where as a function of the signal to noise of the background and the separation of 2 points in PSF, this graph kind of quantifies the fractional error in the center that you locate. And if we're anywhere above a signal to noise of about 10, we do a pretty good job of separating out 2 sources, even if they're only separated by a few PSF. And this is great because the typical signal to noise ratio in these images is around 20. So we're pretty good at separating out different sources. Okay, I've only got a few minutes left, but what did we get at the end of Bolin's project? Well, we're able to run this approach on the data, and we find these transients. Now, you notice a few different things. The first is that there are lots of these transients around the dendrites, and you could immediately just ignore them, but some of them are, interestingly enough, coming sort of lagging a big bright transient. There's some extra diffusion of calcium along the dendrite, and we think that that can actually kind of tell you something about the synaptic events, rather than just thinking that it's complete noise. I should also say that in order to cluster the transients a little bit more than just the images you see here, we actually ran kind of dB scans, so density-based clustering of different points, to make sure that you are not just finding individual speckles. But even in the case of this example, you see that there are some that happen, along the dendrite after the transient occurred. The other thing that you'll notice in this, there are different colors here, and this is where Bolin was separating out transients that had already been discovered by the team, and then potentially new transients that we were found, because we were interested in a comparison between what they had found and what our new method, our new thresholding provided. Okay, so how did we do? Well, here are some examples of our comparison. So the left-hand side is the difference image. The middle panel is the approaches at segmentation or transient detection that they had already tried. And then on the right-hand side is our clean pipeline. These points are already, they're not just individual points. They've already had a little bit of clustering. And so you see that there are some differences. Ours are more localized, and we also are sensitive to, this diffusion along the dendrites. In some cases, we find objects that their algorithm didn't detect. I think that the top image is the most striking, where there's clearly a transient happening, but their algorithm didn't detect it. Although there are other times when we don't detect something that the UNet image does. And so that's where we need to figure out a little bit more tuning of our thresholding as well, because if you see, it's a very faint transient, but it does seem to be there both in the difference image and in, it's detected by the 3D UNet pipeline. Okay, so I'm basically almost out of time, but what's next? So we have got this pretty simple set of steps in our pipeline that allow us to clean images, model the background, detect the transients, and now the next step is a more detailed comparison. Why did we not detect ones that were detected by the 3D UNet algorithm? Why did we detect ones that they didn't? Are there some kind of physical intuitions? But then the next step that we're doing is actually, we're interested in the temporal behavior of the transients, because this is something that they hadn't ever considered before, and using that as another dimension to classify these transients. Because if one is along the dendrite and it has a long tail of brightness, or lots of transients that are related to it, you could think of co-locating those because they have a similar time signature, as opposed to just spatial correlations. So we're really interested in this in terms of anomaly detection. I actually think there's some, we can also apply some of these techniques back to optical images, but this is just another example of how fun it is to take the things that we learn in astronomy and try and apply them to other fields in physics. So thanks.
Well, thanks, Renee, for keeping it perfectly in time. So now we have about 5 minutes for some questions. And, it was a really nice talk. Thank you. You explained so many things in a short time.
I was going quite fast.
No, it was very interesting. You go quite deep and you still give us a good perspective. So any questions? Oh, so I see a process, but any questions? No, I don't have a question. You know, it does happen to be that the images you're dealing with looks like the cosmic web.
Mm-hmm.
But of course, that has nothing to do with the fact that you're using all these methods.
Yeah, I mean, that's
why
she came and spoke to me, because they just look the same. But yeah, the astronomy, we're basically just saying, I think the main thing that we're bringing here in this particular part is that we can do difference imaging. It's not really the actual spatial structure. I am actually interested in additional things we can test, I think, that allow us to help with their other project, which is looking for spatial correlations between the protein structures. But again, those don't look like the cosmic web. So the reason she approached us is actually not one of the things we care about in our analysis. Yeah.
Right. So there's a question from Benjamin Martin.
Great. So yeah, so one of the things that we need to check next actually is, so Bolin has run the calcium clean algorithm on a few of this data chunks that they have given us. And I should say, I didn't state this explicitly, but the data we're using right now are non-moving mice that don't have ALS. We're going to get the other data to run this on first, but we wanted to check in the kind of control. But we really do need to do that statistical analysis, right? So what is our fraction of transients that are real? I think the good thing about our approach compared to the other stuff is that because we can model this pretty well, we can actually also simulate transients that map pretty well onto the dendritic structure. And so we can start to do a much better way of calibrating them or testing for the actual efficiency, as opposed to, in their case, their truth is just what a human labeled. And that isn't necessarily the truth, right? So we don't have an actual number for the efficiency right now, but I think we probably will do pretty well.
There's a question from Gwen and then Kier. Go ahead, Gwen.
Hey. Great talk, Renee. Thank you. I was wondering, is there anything like sort of surprising or interesting that you learned from working with this data that maybe like inspired ideas to bring back to astronomy? Like, has there been any sort of interplay that way?
Yeah, I mean, one thing that I've been trying to think a little bit about is this noise modeling problem. So, we have data that have complicated noise models that, because we can model the physics of an object like a galaxy, we have a sort of analytical description for what we think it should look like. In their case, there's so much variation in the dendritic like shape that they don't model the physics of what a dendrite should look like. It's all empirical. And so the fact that we can't rely on any empirical models has meant we have to do everything in situ. So the background estimation, I wouldn't actually necessarily have thought to do that in an astronomy image, to only use the background and not sort of compute things a little bit more analytically. And so what I'd like to do is take some of this model and take it back to, say, observations that have a lot of seeing, because then you do expect this background to vary a lot more than if you have like deep sky images. So I wonder if it will increase our ability to use kind of what we associate as more, bright sky data. I haven't looked at that in much detail, but I do think there are things that we've learned from the very, very empirical need here that we can take to astronomy where we typically have been relied on modeling.
That's really cool. Thanks, Renee.
Here.
Hi, Renee. I have a sort of related question. As I think you said that you're not seeing the calcium directly, but rather dye that's tracing the calcium. So I thought in some ways that's analogous to astronomy also, in that we don't see dark matter directly, but we see it through traces. Is there any sort of relevant scale-dependent bias model that you might need to map from calcium to dye, or?
Oh, that's awesome. That's really great. I'm not sure about this specific problem, but the other one that I didn't talk about, the synaptic, the distribution of the proteins, there, our colleagues have been trying different dye configurations, and they get kind of vastly different results. And it initially scared me a lot, and they sort of just pick the one that looks the best. But that would actually be really cool to estimate if you could try either different dyes or different bias factors to actually like be able to correct images with different dyes, because typically that's the problem with, they just make choices. I mean, we even ask them to use the microscope at different resolutions to see if we can actually learn about improving the resolution, like doing like adaptive optics, because again, they just fix it. So I think that's a really good question that nudges us in like systematizing all of their assumptions and then correcting for them in future observations. That's great.
Yeah, exactly. Cool, great work.
Thanks.
Okay, I'm gonna cut off here. There may be a bit more time at the end, but thanks, Renee. So now we're gonna have Ting, which you have heard the introduction of. So Ting is our newest faculty member. And Ting, go ahead.
Thank you. I hope you can see my screen now. So it's a pleasure to give a talk here. And for those who don't know me, I'm Ting Li, and I actually in my office now. And although I don't have any book in my bookshelf, but I hope I'll get them soon. And so I just joined the U of T over the past summer, and I'm very excited to be here. And I want to just show you guys with the next 20 minutes about the work I've been doing. In particular, I'm going to show a survey that I'm leading that called the Southern Stellar Stream Spectroscopy Survey. And I'll talk about more of these later, but what you're seeing is the distribution of the 12 streams that we followed up spectroscopically, and I'll show more results from that later. Okay, so for those, especially for students, just want to give a very brief introduction about the background. So the Milky Way, like galaxies, are assembled by creation and disruption of many small systems. So it's something, small system like dwarf galaxies or globular clusters, when they orbiting around the Milky Way like system here, they got tidally disrupted. And before they got fully mixed to the Milky Way stellar halo, one of the process in the middle is this long, thin streams, and that's the subject that I want to show for today. So these kind of stellar streams are actually seen in the observations. So what you are seeing here is the one of the map from the Sloan Digital Sky Survey in the northern sky. So this is basically a stellar density map, but color-coded for different distance. And you can see these kind of thin structures, that GD1 stream, Power 5 stream, Orphan stream, Sagittarius stream. So this was something discovered with the Sloan Digital Sky Survey. And here is like another patch of the sky in the southern hemisphere that covered by the dark energy survey. So another optical imaging survey. Again, the different colors sort of like a different distance, and you can see these kind of thin structures, and that's the subject what I would like to talk about. And recently with the Gaia release, like there's also a lot of stream discovered, especially within the inner part of the Milky Way's halo. So this is a map showing the streams identified within about 12 kiloparsec. So some people make joke like, oh, our Milky Way stellar halo is full of spaghetti. So it's just like long structures on the sky that either disrupted globular clusters or dwarf galaxies. So I think there are a lot of science that we can do with stellar streams. I'm kind of trying to highlight in 3 different aspects, and I'm trying to propose the science related to this with like what kind of question we can address or answer with stellar streams. So first is the formation of the Milky Way stellar halo. The question we can ask is like, what are the building blocks of the building blocks of the Milky Way stellar halo? Where do the most metal pool stars come from? And so stellar stream is kind of in the middle of the phase between intact globular cluster dwarf galaxy and the fully mixed Milky Way's halo. So that's hopefully can help us answer a question like that. And the middle panel here showing the questions about the Milky Way's mass and potential. So the question we can ask is like, what is the Milky Way's virial mass? It's 0.8 or 1.6 times 10 to the 12 of the solar mass. This is a plot showing like the Milky Way mass measured by different group using different tracer. You can say you can see that depend on which group is doing the work and using different tracer, there's a factor 2 to 3 between 0.7 to 2 times 10 to the 12 solar mass for the mass of the Milky Way. And the other related question is the Milky Way's potential is spherical or triaxial. And then the third component, I think slide stream would be interesting to answer is a question like, is dark matter cold or warm or self-interacting? So the plot here showing is just a simulation with a cold dark matter CDM or warm dark matter WDM. And you can see the number of subhalos are expect to be very different with different dark matter models. So this is something you might have heard about missing satellite problem. And hopefully that we can use to stream to do a study on this. In particular, we know that these subhalos, some of the subhalo here will form galaxies, and we can observe these as satellite galaxies of our Milky Way. But there are also certain subhalos that are much lower mass, say below 10 to the 7 solar mass. And the question is, these galaxies will never form stars inside. Can we actually find them and confirm the existence of those? I think that's also something the stream can probably help us to answer. So with all of those kind of interesting questions in the science, I started a program called S5, the Southern Stellar Stream Spectroscopy Survey. And I started this in the summer of 2018. And really it was started because of a lot of stream that we discovered in the dark energy survey, plus the release of Gaia DR2, that we can use all the greatest photometry plus a proper motion to do very efficient target selection. In the meantime, we use the telescope in Australia. So this is a 3.9 meter telescope in Australia, Angular Australian Telescope, AAT, which has a 2-degree field with 400 fibers that you can observe 400 targets at the same time to feed to this spectrograph. So we're able to cover a lot of streams on the sky over a wide area. And then we select the member stars we find in these streams and then observe these brightest stream members with the Magellan telescope in Chile, a 6.5 meter telescope. So that's bigger. And then we can get this single object shell spectroscopic data with a microspectrograph. So here we get the kinematics, and on the right, we get the chemical composition of those stars, which give us a lot of information. And we have published so far 9 papers, I think, accepted, and 1 paper in the review. I won't have time to go through all of them, but I want to come back to this paradigm and just show a few highlights of the work that we have been doing. So again, it's the formation of stellar halo, the Milky Way mass, and the dark matter subhalo mass function. And I'm going to pick one highlight from our work for each topic. So for the formation of the Milky Way halo, so one thing we discovered with our spectroscopic data is we find a stream called Phoenix stream. Its metallicity is lower than any of the known globular clusters. So the plot, what you're seeing here is a distribution of the globular clusters in our Milky Way in metallicity. So you may see that all of the globular clusters are all have metallicity higher than -2.4, which sometimes we call it as like metallicity floor. And we haven't found any globular cluster that have a lower metallicity than this. So the Phoenix stream, which we believe the progenitor is actually a globular cluster. But when we go measure the metallicity from our observation, we find that the star will have a metallicity below -0.2.5. Actually, the mean metallicity is -2.7. So we think we find a case that in a stream that it's originally was a globular cluster, but now get disrupted and has a much lower metallicity than any of the known globular cluster. So this is either saying, okay, these kind of globular clusters has formed in the past, but just not seen because they are disrupted. And maybe we also will be able to find more such systems in the future, but that's very exciting. And in terms of the middle panel that I was showing earlier is about constraint of the Milky Way mass. So that's something we are currently working on, but we also find that in order to constrain the mass of the Milky Way and the potential of the Milky Way, we really need also to understand the impact from the larger Magellan Cloud, which is the largest satellite galaxy of our Milky Way. So here is a work that we did with 5 stellar streams that observed with S5. For each of them, we use them to constrain the mass of the LMC. And so you can see that each different one we use to constrain the LMC independently. And then we got a very similar result or consistent result. So we think the LMC mass is something between 1.5 to 1.9 times 10 to the 11 solar mass. And also, in order to fit the data, we require a lighter Milky Way mass around something like 0.8 times 10 to the 12. So actually, you will see that LMC actually is about 20% of almost 20% of the mass of the Milky Way instead of the 10% that what we thought in the past. In terms of the subhalo mass function component, what I'm going to show you here is something we also find using the survey data that we have been doing. So originally, here are 2 streams. One is called Alika-Uma stream, another is called Atlas stream. So we have these 2 streams that were discovered in the dark energy survey and as Atlas survey in the past. So we thought those 2 are 2 separate streams. But when we follow up them spectroscopically, here what's showing is the velocity as a function along this direction in phi1, so stream longitude. So you can see in the velocity, these 2 streams are seamlessly connected. Actually, not just velocity, but also the proper motion from Gaia, the distance from the distance tracer, all showing that they are seamlessly connected, although there is some kind of discontinuity on the sky. So this kink or spur, that what we call, is very similar to the one we see also in GD1 streams, another one showing similar signature. That's possibly it is caused by a perturbation from a dark matter subhalo. So it is very interesting to get more data in this area and then try to see what are the mass of the perturber and the orbit of the perturber, and if it's associated with any of the baryonic satellite, globular cluster, or dwarf galaxy, or it might be just a dark matter subhalo. Yeah, so this is what we have done with S5 data on stream-related science. But I also want to emphasize, especially to this group, because not everyone's interested in science with stellar streams or like near-field cosmology. So I want to just highlight a few other things we have been achieving using the S5 data. So this actually the first result come out from our collaboration before any of the interesting science result from stellar streams. So after the first internal data release, we find a star with a line of sight velocity about 1,000 kilometers per second and a 3D velocity about 17 kilometers per second. It's the fastest main sequence star ever found. And most exciting part is when we rewind its orbit, because we get all the 6 information, we found it's actually coming from the center of the galaxy. So it's really coming from ejected by the supermassive black hole in the Milky Way center. And more excitingly, that if you calculate the ejection velocity, this is the star that we find, while all the other previous known hypervelocity star have a much lower velocity compared to the one that we find. So this is more related to like still Milky Way, because it's a star we find in the Milky Way. We use the sphere fibers to target the Milky Way halo star. And actually, when we also realize that we have a lot of sphere fibers in this program, so we are using them to target low mass, low redshift galaxies. So that's the S5 LOSI component. We have not have any paper published that yet because we don't really have enough manpower to work on that, but I think that's another exciting component. So we're trying to target a lot of low redshift galaxies, like LMC-like, Fornax-like galaxies within the local group and local volume. And this is what we have been getting in terms of the redshift for all of these southern galaxies we have observed. And hopefully this will be a good data set to understand the star formation history of these low mass galaxies, and also maybe provide a sample for photo Z calibration in the future for nearby galaxies. Okay, so what I've showed in the past slide is something probably you have heard before, because it's something we have published. But here I want to use the last 5 minutes to just show you a work that I've been working on. Hopefully will be on archive next week. So early this year, I realized, oh, we have already observed 12 streams that has no progenitor. So we call progenitor-free stellar stream. That means it's not like Power 5 stream that we see a globular cluster in the center and then tidal tails around the globular cluster. It's more like GD1 streams. Basically, we don't recognize any of the progenitor, but we see this kind of structure on the sky. So S5, I've already observed 12 streams, and I said we should do some chemical, orbital and chemical property study on this stream. So we took these 12 stream and made some study, and this is a paper in preparation that hopefully will be submitted within the next week. So the first thing we did is to take a look among these 12 stream. Oh, one thing I wanted to highlight. So maybe if you know how to count, right, you will see 1, 2, 3, 4, 5, 6 here, but then there's 1, 2, 3, 4, 5, 7 here. And we are not considering Sagittarius stream here because it's a stream we all know a lot of people has observed in the past. So you will say that's 13 stream, not 12. I just wanted to make it clear that so the Chenab stream here in purple and the Orphan stream here in orange are actually one stream, although there's a long story about how this was discovered, why there's two names. And here we separate them into two portions with different color. We just want to see if there's any changes. That's why we made that. But in total, it's really the 12 stream that I'm going to talk about. So the first thing we did with these 12 streams to study what are the progenitors. As I said, the streams can be disrupted dwarf galaxies or globular clusters, but really we didn't have a chance to really look at them like more with a larger sample. So this is what we get. So we find among the 12 stream we have, 6 of them or half of them has a very low velocity dispersion and a very low velocity dispersion, which indicating their progenitors are more likely to be globular clusters. And throughout the plot, if you see different plots, you will see they are indicated as a circle. And for the other half of the stream, they have either a larger velocity dispersion and high velocity dispersion. So we think these are the dwarf galaxies. So in our 1,000 stream sample, about half of them are globular clusters and half of them are dwarf galaxies. That's the first question we wanted to know from, we would ask from our sample. The second question is, what are the luminosity and the stellar or the stellar mass of these stream progenitors? So it's hard to say this because it's really hard to count every single stars because these are disrupted system. It's hard to count every single star to know what are the luminosity or the stellar mass of the progenitor. But for the dwarf galaxy stream, we can use some empirical relation like the mass velocity relation. Based on their mean velocity, we can go get what the luminosity would be. So for all the dwarf galaxy stream, which the ones indicated in cross, we kind of can map them to see what are their stellar mass or say the luminosity are. So for these 6, we see that the luminosity is about between MV of -5 to -10. This is what we are getting. So give you an idea. So this is where we separate between the classical dwarf galaxy like LMC, Sculptor, Fornax, and ultra faint dwarf galaxy, like the faint ones that we're finding recently a lot with the modern imaging survey. So the one that this stream progenitor with the dwarf galaxy stream sample, they're really kind of at the lower end of the classical dwarf and the higher end of the ultra faint. And let's take a look about the simulation, what the simulation predicts. So this is the table taken from this paper from the Fire 2 simulation. So they calculate the number of streams at a stellar mass higher than 5 times 10 to the 5, which is around roughly at MV of -9.5. So this vertical red line here. So they predict among their 12 simulation, there's about 3 to 13 streams in this regime. So in this end, but for what we know in our Milky Way, only one stream actually is in that mass range, which is a Sagittarius stream. But this is a big kind of discrepancy between what we have seen here and what the model predicts. So you might have heard too big to fail problem in dwarf galaxies, which is saying the simulation predicts much more massive galaxies, dwarf galaxies, than what we have seen in our Milky Way. And actually, this is probably also a problem now in the stream. Basically, the simulations from this simulation, it predicts way more stellar stream in the massive end than what we have known so far. But it's possible that so I think we should try more different simulation. Hopefully there will be more simulation to check this, and then maybe there are more massive ones that just haven't been discovered in the so far, but that's definitely an interesting thing. Another thing we look at is the orbital property. So here I'm plotting is the energy versus angular momentum for all the 12 streams that we have observed. So if it's close to 0 in angular momentum, it's in a polar orbit. If on the right is a retrograde orbit, if it's on the left is prograde orbit. You can see the distribution of the prograde versus retrograde is not very uniform. So this is where our disk is located at. This is another structure that you may have heard called Gaia Encelada sausage. So you can see there is a much higher preference in the prograde orbit than the retrograde orbit from this 1,000 stream. And let's take a look about again what the simulation says. So the fire simulation from the same paper, they predict a uniform distribution in the orbital poles. There is not a preference between the retrograde and prograde orbit. I think this is very interesting. I don't have time to go do more on this, but there's also very interesting thing on the classical dwarf. There is a very well-known thing called a V-pulse structure. So the dwarf galaxies in the Milky Way also show some preference in the prograde orbit than the retrograde orbit. Okay, so the last point I wanted to show is on the globular cluster stream. So I talk a lot about the dwarf galaxy stream, about their progenitor mass, etc. But I also want to highlight one result from the work showing about the globular cluster stream. So we have 6 globular cluster, and what here we are plotting is eccentricity of the stream and also the mean velocity of the stream. And also plotted are the globular clusters in our Milky Way. What you are seeing here in the stream part, I think is very interesting relation for the eccentricity and the metallicity, this strong correlation that is saying the higher eccentricity globular cluster stream also have a higher metallicity. And also the majority of our globular cluster stream have a much lower metallicity compared to the intact globular cluster. And one thing I want to highlight is this Phoenix stream that I mentioned earlier that we find as the most metal pool globular cluster. So it's not only the most metal pool compared to all the known stream and also the known globular cluster, it also has the lowest eccentricity. So because it's on a circular or quasi-circular orbit, it might be the reason explain why it has so low metallicity. Basically, as it's on a circular orbit, it can have a longer lifetime for dissolving the globular cluster to be fully mixed. So that's why maybe the reason we are still seeing it with such a low metallicity. And another globular cluster stream I want to highlight is this one. So this one is called the 300S stream, and you can see at a metallicity about -1.2 and at eccentricity about 0.8. So this high eccentricity, a bit high metallicity, I don't know what you can connect to link, but I immediately thought about the Gaia Encelada sausage, which is at a similar metallicity and also at a high eccentricity. And let's take a look. This is where 300S is at. So it's also on a polar orbit. So we think that the 300S probably is a stream, a globular cluster. Its progenitor is a globular cluster of Gaia Encelada sausage, or maybe it's a nucleate, the core of the Gaia Encelada sausage. I think we need more data to prove this in the future. But this is just a, yeah, that's what I'm saying. But this is just a very tiny part of the paper. There are a lot of other things discussed in the paper, and I hope it will be on archive next week. And I encourage you guys to take a look if you're interested. But I want to end my talk with a great list of people that from the team that without them, all this awesome work won't be done. And I also would hope that now I'm at U of T that I will have more people interested in this and collaborate or working in S5. And that's all for today's talk. Thank you.
Well, thank you. It's a very fast-paced talk and very exciting science. So any questions from the audience? We have about, we'll give you 5 minutes to discuss the talk. Let me start with a question. You show this eccentricity and metallicity, kind of a scaling or kind of correlation. Shouldn't that, I guess I didn't quite understand it. Shouldn't that, instead of eccentricity matters, shouldn't it be the pericenter distance that tells you the disruption time? Or?
That's also related, I would say. We have some study about the pericenter of all the streams and how that connects. I think this might be one of the reasons that the the eccentricity might explain why we still see this stream, because there are some papers discussing about how the dissolution time is related to the eccentricity of the orbit. But I agree that the pericenter definitely matters. These streams, oh, I forgot to say another thing for these 12 streams, they are all farther than 10 kiloparsec from the Milky Way. So they are not those stream that has been found in for example, the Gaia EDR3, which are mostly in the halo. The one that I'm showing is all between 10 kiloparsec to 50 kiloparsec. So for example,
I see.
Most of the stream here has a pericenter, I think less than 8 or 7 kiloparsec. Most of them are never get within like 8 kiloparsec, yeah.
So this is kind of a survival plot. This stream still survives, or still look like a stream because
I think that's very interesting. I think that's a very interesting question, like ask why this stream, for example, is still surviving, and that will be interesting to see what the theorists think about. But I think there are questions in terms of like whether these streams are created earlier because they are low mass velocity, while these streams are created recently because they are higher mass velocity. I think there's all these kind of discussions on that.
Thank you. So any other questions, please? We actually have a lot of local experts on dynamics and galaxies and streams. Any students want to ask a stupid question? This is your chance because we don't have any other question at the moment. Well, let me also make the comment that all the astronomy 425 students and 424 students are sitting here listening to all these astronomy talks, and they're looking for potential projects, potential supervisors, and I think Ting has offered a large number of projects to the students. And, well, now you see who she is and go talk to her. Okay, I don't see any other questions, so let's give a silent applause to two dynamic speakers we have today, and we'll see you next week. Thank you.